Today, we will discuss Amplicon sequencing and how to assess the success of our Amplicon sequencing. First, we'll review some general information about Amplicon sequencing, then library preparation, sequencing, and sequencing run review, analysis files, and reviewing analysis results. Then, we'll cover some additional resources for Amplicon sequencing. I will link to these additional resources and several documents along the way for more in-depth information on all of these topics. We will be covering quite a bit of ground today, so I do encourage you to keep the resources in this presentation for additional information. Amplicon overview. What is Amplicon sequencing? Amplicon sequencing is a type of targeted sequencing. So instead of sequencing complete genomes or whole transcriptomes, we are sequencing only a portion of the DNA or RNA from our samples. Amplicon libraries are typically prepared using a PCR step to selectively amplify the regions of interest, though there are some other library preparation approaches as well. With Amplicon sequencing, we can assess germline variants, so genotypes, or somatic variants. Why would we choose Amplicon sequencing instead of whole genome or whole transcriptome sequencing? With Amplicon sequencing, we can sequence just our targets of interest at greater depths, which gives higher sensitivity. Amplicon sequencing is often faster and more cost-efficient, and Amplicon sequencing is highly versatile. It can be used for many types of samples and a broad range of applications. There are two main approaches to Amplicon sequencing. The first is single Amplicon sequencing. This is where we target a single locus. This approach is most commonly used for metagenomic studies and can also be referred to as metabarcoding. Two applications of this are the bacterial 16S sequencing and fungal ITS sequencing. The second is Amplicon panel sequencing. Amplicon panels include multiple loci of interest and can be used to sequence full genes, genomic regions, or targeted SNPs and indels. Panels are used in many contexts, including research, genotyping, oncology, pathogen sequencing, and others. Today, we'll be focusing on Amplicon panels for our discussion. For more information on single Amplicon libraries, sequencing, and data analysis, we have a two-part metagenomics webinar series, which discusses the full workflow for metabarcoding studies. These webinars can be viewed at any time with the links here. Now let's take a look at Amplicon library preparation. In Amplicon library preparation, we use primers to amplify the region of interest. Input can be DNA or RNA, though for RNA, we will convert the nucleic acid to cDNA prior to target amplification. After we amplify the target regions, we will add adapter sequences, which are required for compatibility with Illumina sequencers. Here, we will review two methods for Amplicon library preparation, AmpliSeq for Illumina and extension ligation-based prep. These are the two chemistries used by Illumina library prep kits for Amplicon library preparation, though under vendors or published protocols may use alternative workflows. We have a fantastic webinar, Amplicon Sequencing Introduction, which reviews Amplicon library preparation, sequencing, and run review in much more detail than I will today. If you did not attend this webinar when it was given live, I do recommend reviewing the recording if you would like more information on these topics. In the coming slides, I will give a high-level look at library preparation and sequencing, though again, not as much detail as in this webinar recorded here. We will start with AmpliSeq for Illumina. In this workflow, we use multiplex PCR to amplify our targets of interest. After this, we do adapter ligation and additional PCR amplification before sequencing. AmpliSeq panels can be used to target a wide range of regions from 12 amplicons up to thousands. Input can be DNA or RNA and can be high quality or degraded, including from FFPE. We have a selection of panels with pre-designed content for various applications, and we offer custom panel designing to meet the needs of nearly any project, including for non-human species. We have two flavors of pre-designed AmpliSeq panels. The first is off-the-shelf panels, which have been fully tested and validated by Illumina. We provide specific recommendations for data needs, read lengths, and more. We also have community panels, which are panels designed by independent experts in their field. These have not been fully validated by Illumina, and they can require additional optimization. Here, I have listed the pre-designed DNA, RNA, and DNA and RNA panels. I will link to a full list of all the community panels that we offer a little bit later in the presentation. Let's briefly review the AmpliSeq for Illumina library preparation workflow. And we'll be starting at the top of the image here. So we start with our DNA or RNA. If RNA, we first perform reverse transcription. We move forward with our DNA or cDNA and perform the multiplexed PCR with the PCR primer pairs targeting our regions of interest. Once amplified, we remove the locus-specific primer sequences from the Amplicons. Then we ligate the sequencing adapters and further amplify the libraries prior to sequencing. I've included links here for more information on AmpliSeq library preparation, panels, and custom panel design. So now we'll go ahead and touch on extension ligation-based library preparation. With this method, we use single-stranded probes that bind to the DNA or cDNA. Then we perform an extension ligation reaction between the target probes to create a single-stranded amplicon. After this, the sequencing adapters are added via PCR. This chemistry is used in the TrueSight myeloid library preparation workflow, which creates libraries for researching myeloid cancers. And it is also used in the TrueSeq targeted RNA expression library prep kit, which allows for custom RNA panel sequencing. In this workflow, starting from the top again, pre-designed probes are hybridized, flanking the regions of interest in the sample. A polymerase extends from one of the probes, and we ligate the extended region to the second probe, giving us a single-stranded amplicon. We then use PCR to add the adapters to create our sequencing-ready libraries. More information about the TrueSight myeloid and TrueSeq targeted RNA expression workflows are available at the links here. Now that we have our libraries prepared, let's go ahead and proceed with sequencing. Before we sequence, we do need to answer a few questions that will dictate how we proceed with sequencing. We need to decide where the sequencing will be performed, which sequencer to use, the read lengths for the sequencing run, the number of samples that will be pooled per run, and the percent of the PHYX controlled library we will add into the sequencing run. Where to sequence? You may have the sequencer that you would like to use in your lab. If not, there are sequencing service providers that can perform the sequencing for you. These can be academic or commercial facilities. If you would like assistance connecting with a sequencing service provider, please feel free to reach out to tech support or to your local sales team. Which sequencer to use? Illumina offers many sequencers based on the type of sequencing performed and data needs. Amplicon libraries are often sequenced on the benchtop sequencers, which are shown in the top row of the slide here. So that's the iSeq, MiniSeq, MySeq, and NextSeq. The higher output sequencers on the bottom row, they are compatible with Amplicon sequencing. They're often not appropriate as they give more data than is typically needed for most Amplicon applications. When we get to sequencing, we want to ensure that we are using a suitable read length for the libraries. Amplicon libraries can have different sizing depending on how the panel is designed. Longer Amplicons will require longer sequencing reads, and shorter Amplicons will require shorter sequencing reads. Illumina provides recommended sequencing read lengths for all of our library preparation workflows, and tech support can also provide read length recommendations for other library types as well. Next, we have to decide on how many samples we can pool per run. And there are two main factors that will help us to decide. The first is the number of index sequences. All samples pulled together must be uniquely indexed. So if you have 96 indexes total, the maximum number of libraries you can pull per run is 96. The second factor is the data needs. This refers to the number of reads you would like per sample, and is also called the coverage. Data needs depend on the panel size, so larger panels will require more data than smaller panels, and the application. For example, somatic variant calling requires higher coverage than germline variant calling. We do provide read length and pooling recommendations for pre-designed panels, including AmpliSeq for Illumina panels and TrueSight myeloid panels. These recommendations are featured on the product pages for each library preparation kit. Please feel free to reach out to tech support if we can assist with panel recommendations. Recommendations for custom panels will depend on the specific panel, design, and experimental goals, and this applies to custom AmpliSeq panels and TrueSeq targeted RNA expression libraries. Phi-X-Spiken is our last step for run planning. For the vast majority of cases, we recommend spiking in Phi-X at 1% as a sequencing control. If you anticipate low base diversity or skewed base diversity, you can increase the Phi-X-Spiken to provide additional base diversity to maximize run performance. The percentage of Phi-X in these cases will depend on the sequencer used. In these cases, please don't hesitate to reach out to tech support if you have any questions for your specific panel. Our first step in assessing the success of our Amplicon sequencing is to review the sequencing run performance. We want to ensure that the sequencing performs well and that our data is good for downstream analysis. For this, we're going to focus on two key sequencing metrics: the %Q30 metric, which is a measure of the data quality, and the data output, which is also called yield. Illumina provides specifications for each sequencing kit for all sequencing platforms. This is where we can look to see the expected Q30 percentage and yield for the sequencing kit we are using. In this example, for the mini-seq, we can see the expected output or yield values for each sequencing kit in gigabases. Looking lower on the page, we can see the expected Q30 percentages for each kit read length. We have two options for reviewing our run performance. The first is in Base-Based Sequence Hub online, and the second is the Sequencing Analysis Viewer Software, or SAV, which is an offline software. Today, we will review an AmpliSeq for Illumina myeloid run in Base-Base, so I will also include resources for SAV a little bit later. Overall, both are structured very similarly and give essentially the same information. In today's run, the pool we'll be reviewing is just DNA libraries, and the pool was sequenced on the MySeq with version 3 or V3 sequencing reagents. So first, let's take a look at what our project recommendations are. Here, we are looking at the project recommendations on the product page. We want to look at the section for the MySeq. Next, we know that we are using the MySeq version 3, V3 reagent. And then we are sequencing DNA-only libraries. So we see that we can pull 12 samples per run, and we want to use a 2 by 151 base pair read length. Now, let's take a look at the specifications, so we will know what metrics to expect when we're reviewing the run. We are using the MySeq V3 reagent, and we are using a 600 cycle kit. Now, in our run, the optimal read length is 2 by 151 based on our libraries, so we are not using all of the cycles in the sequencing kit. This is perfectly fine, as we always want to make sure that we are using an appropriate read length based on the libraries we are sequencing. So we will need to do a few additional calculations to account for the shorter read lengths. Indicated on this slide is the yield or expected output for a 2 by 300 base pair run. We are using a 2 by 151 base pair read length, so about half. With this, we will want to half the yield accordingly. So halving this gives us an expected output of 6.6 to 7.5 gigabases. Now let's take a look at the quality we can expect for the V3 run. Again, since we're using the 2 by 151 base pair read length, we need to do some calculations. We can also compare the V3 read quality to the V2 reagents, which do have a standard 2 by 150 base pair configuration. Taken together for this run, we can expect our Q30 percentage to be at least 80%. Now let's move on to taking a look at the run. When we open up the run in BaseBase, we'll first be shown a summary page. To review the run performance information, we want to select the metrics option near the top of the page. This will take us to our metrics screen. Here, I have scrolled down the page slightly to the per read metrics table and enlarged the table so that we can review it more easily. First, we can check our yield. So in the yield column, we can go all the way to the bottom to see the total yield for the run. Here, we see it as 8.69 gigabases. From the specifications page, we are expecting 6.6 to 7.5 gigabases, and our yield of 8.69 gigabases exceeds this target, which is absolutely fantastic. So this lets us know that our yield for the run is high, and it exceeds our expectations and specifications. Second, we will evaluate the data quality. We will look at the percent Q30 column and then look at the bottom of the table for the run total. In this run, the percent of the data generated that is at or above Q30 is 94.92%. We wanted at least 80%, so we also exceeded our quality expectations for the run. Reviewing the run performance is our first step in assessing the success of our Amplicon sequencing. For this run, we are exceeding both our output and quality specifications, so we know that the sequencing run performed well and we can proceed with data analysis. The first 4 links provide additional information on the different sequencers, including the specifications for each sequencing reagent kit and sample pooling. The last link leads to our BaseBase public data. We have public data from many AmpliSeq panels, including run performance and analysis results, and the public data is a super helpful resource to let us know what we can expect for various panels sequenced on the different sequencing platforms. More information, including the download for the Sequencing Analysis Viewer software, is here. We also have a helpful recorded webinar for how to use Sequencing Analysis Viewer and review run metrics available on our webinar page. The support bulletin at the last link also reviews steps for run evaluation. Now that we have seen that our sequencing run performed well, we can move forward with data analysis for our Amplicon libraries. First, to give an overview of Amplicon data analysis. Our first step is to generate the FASTQ files from the raw sequencing data. Then, we'll remove the locus-specific primer sequences if they are present in the data. We will align the read to the appropriate reference sequence. This is usually a reference genome based on the species, even for RNA samples. We count the reads aligning to each amplified region to understand the coverage of each target. We characterize the variants, so any bases or regions that differ from the reference sequence. This step includes detecting SNVs and indels. And then for some RNA-based samples, we will assess reads for evidence of RNA fusion events. We have different options for Amplicon analysis depending on the library preparation chemistry and where you would like to perform the analysis. For AmpliSeq for Illumina, we have the DNA Amplicon, RNA Amplicon, and the combined DNA and RNA Amplicon analysis workflows. And these are available both in BaseSpace online and offline via local run manager. We also have the Dragon Amplicon app, which is available online in BaseSpace and offline on the Dragon server. For extension ligation-based data, we have the TruSeq Amplicon analysis for TruSight myeloid data and the TruSeq targeted RNA analysis for TruSeq targeted RNA expression data. Both are available in BaseSpace and within the MySeq Reporter framework. Today, we will review the results for AmpliSeq data analysis with the DNA Amplicon app, as that is the most common workflow used for Amplicon sequencing and data analysis. To analyze our Amplicon data, we will need three things: our FASTQ files from the sequencing run, our panel manifest, and our reference genome. FASTQ files list all of the sequences, the A's, T's, C's, and G's, sequenced on a per sample basis. Depending on how we are setting up the sequencing, FASTQ files may contain 100, thousands, or millions of reads for each sample. FASTQ files also include quality scores for each and every base. Depending on the analysis workflow you are using, the FASTQ generation may be incorporated, or you may need to generate the FASTQ files in advance. The next thing we need is our manifest. A manifest is a .txt file which lists out all of the targets in the panel we are sequencing. For pre-designed panels, Illumina provides the manifest for download on the support page for each of the panel kits. Manifests for custom panels can be downloaded within the Design Studio portal by the panel owner. Manifests are .txt files, so just plain text files, so I'd like to view them in Excel instead of a text editor, as it's easier to see the proper formatting, like how the columns align. Manifests have three sections: the header, which includes basic information about the panel and the manifest file; the probe section, which lists out gene and panel targets and the sequences used to target the regions of interest; and the target section, which includes genomic coordinates for the targets, the DNA strand designations, and the reference sequence for the amplicon. In the header, the most important sections to note are the manifest name, which lists the panel, and the build ID, which references the correct reference genome to use when performing analysis. When we move to the probe section, starting at the left, we see the target ID, which lists the unique names for each of the amplicons. Moving toward the right, we see the ULSO and DLSO columns, which are the upstream and downstream locus-specific oligos, so the locus-specific primers for each amplicon. Last, we see the gene ID. We often have multiple targets for each gene, allowing for coverage of all or larger parts of genes. Scrolling down further, we get to the target section. Again, from left to right, we have targets A and B, which are the same for most panels. We have our target number. Intended panel targets will have a target number of 1. Known off-target amplification products will have a number higher than 1. Then, the chromosome start position and end position gives us the genomic coordinates of the target amplicons. The probe strand references the genomic strand targeted by the primers or probes, and the sequence gives us the reference sequence for the amplicon target. Manifests for some RNA panels are encrypted and will be .emf files. While these files cannot be opened and viewed like the DNA manifest we just reviewed together, they can be used for analysis with the Illumina analysis workflows the exact same way. All right, the third thing we need for data analysis is our reference. Here, we usually use a genomic reference, even if data is generated from RNA samples. Illumina provides human references, as well as other species, for use with Illumina analysis pipelines. It's also possible to create custom reference sequences if we do not have a pre-prepared reference. Genomic references are available on the iGenomes page linked here. Also, references are usually super big files, so the downloads will be zipped. It is important to unzip the reference files before use and to start the download well in advance of when you'll be needing it. Now that we have the files needed to start our analysis, let's review the results of our analysis. First, we will focus on alignment metrics. Our first metric is alignment, so how well or what percentage of the reads are aligning to the reference genome. Then, on target. This metric tells us what percentage of the reads are aligning to the targeted region. Coverage refers to the sequencing depth or reads per amplicon. Dropouts refer to amplicon targets that do not receive any reads. And uniformity is a measure of how even the coverage is. So this takes into account the coverage for all of the panel targets. Let's quickly visualize what each of these metrics mean. So first, we need a reference genome. The genome will be used as our reference sequence, to which we will align our sequencing data, our reads. Here, I've added reads to our schematic. Aligned reads align to the reference sequence. Unaligned reads do not align to the reference sequence. On-target reads align to regions defined in the manifest file. So these are the regions we want to sequence with the panel that we're using. Off-target reads still align to the reference sequence, though they do not align to the regions listed in the manifest file. So these are reads that are not within the target area of the Amplicon panel. Coverage refers to the number of reads for each panel target, or reads per Amplicon. Amplicon targets in the manifest that do not get any reads are known as dropouts. For our coverage uniformity calculation, we look at the reads for all of the Amplicon targets and compare them. And this metric will be calculated for you during analysis. For us to review the analysis results, we'll need the analysis output files. If the analysis was performed within Local Run Manager, the output files will be in the alignment folder in the analysis output location. If the analysis was performed in BaseSpace, the analysis files will be in the Files section of the BaseSpace project, and I will give more information about this shortly. If you have any questions regarding where to find analysis files or how to interpret them, please reach out to technical support. For reviewing analysis results, we will continue with the AmpliSeq DNA myeloid libraries, for which we reviewed the sequencing performance. So before we review our analysis results, we want to know what we can expect. For pre-designed panels, we do provide expected metrics in the datasheet for each panel. Each of these datasheets are available on the respective product page. These are not specifications like we reviewed for sequencing run performance. Rather, these are the metrics we commonly see for each panel. Depending on the run setup, input, samples per run, and other factors, these metrics can vary. For custom panels, expected metrics will need to be tested and evaluated and the performance optimized. Depending on the panel content, alignment metrics can differ from pre-designed panels, and that is totally fine. There's a lot of helpful information about each panel in the datasheets, though for now, we'll be focusing on just the alignment metrics. As we review the metrics in the coming slides, I'll remind us of the metrics listed in the datasheet. First, when you open the analysis, you will see a summary of the analysis results. These are high-level and list the results for all of the samples included in the analysis session. You can also review summaries on a per-sample basis, though here I will review the summary results for all of the samples. This is a quick way to evaluate many of the metrics for all of the samples analyzed and helps us to compare the performance of the different samples with each other. Scrolling down this page, there are several tables, and I will review them in the coming slides. The first table I'd like to review is the read level statistics. To the right of the table title, there is a little eye icon. Whenever you see this icon, you can select it, and it will give you additional information about each of the metrics, what they mean, and how they are determined. So here we see our percent aligned reads. So the reads align to the reference genome, and they are all at about 95%. We did not have a value for alignment in the datasheet, so 95% is very good. Next, we will look at the percent on target. So the percentage of the reads that are aligning to the regions defined in the manifest file that we use for analysis. Another way to think of this is the percentage of the reads that are going to the targets that we want to sequence with the panel. So here, we see a range from 87% to over 95%. In the datasheet, the ideal minimum was 80%, so our on-target alignment looks fantastic. In the coverage summary table, we can review the average Amplicon coverage and the coverage uniformity. The target mean coverage depth we want depends on the application and the panel. For the uniformity, the datasheet lists 90% as our target minimum. Here, we see the samples have a uniformity between 97 and 99%, so we are exceeding the 90% minimum. This lets us know that the coverage for the different amplicons in the panel is very even. We have now completed our review of the high-level alignment metrics for our samples, and everything looks great. So now we can proceed to the third and final step of our assessment, reviewing the sample-specific results. For us to review the sample-specific results, we first need to select the Files option near the top of the analysis page in BaseSpace. This will bring us to a list of all of the samples included in the analysis. We can select the sample we want to review. This will take us to a list of all of the analysis output files in the sample. So all of the files you see listed here are specific to just the one sample that I selected. In the next several slides, we'll go ahead and review the content and purpose of these output files. First, let's talk about the PDF report. This is a multi-page PDF report that reviews sample-specific metrics. It includes many of the same metrics we've already reviewed when we reviewed the summary of all the samples in the analysis. So here, it is in a PDF format for a single sample. This document also includes additional visualizations for some metrics and an overview of the variants identified in the sample. Clicked a little too fast. Sorry about that, guys. All right. So since we already reviewed many of the metrics in the report, in the analysis summary, I will not review them again here. I do, however, want to call attention to the coverage summary portion of the PDF report. Here, we see the mean Amplicon coverage and the Amplicon uniformity metrics for this sample, which we already reviewed, but we also have a helpful coverage plot for visually reviewing the coverage of all of the Amplicon targets in the panel. The PDF report also lists high-level information about the variants found in the sample, including types of variants, variant locations, and the consequences of the variants. If we go back to our list of files, there's also a summary.csv file. This file also includes the high-level metrics we have already reviewed, but in a .csv format. Two key files in the output are the BAM and the BAM index file, or BAM.bai. These are some of the primary output files of the alignment to the reference. We can use the BAM and BAM index files to review how our data aligns to the reference. The image here is from IGV, the Integrated Genomics Viewer software, though there are other programs that allow you to view these files as well. With the BAM files, we can look at exactly how the data is aligning and see the positions of the variants. The next file we can review is the coverage.csv file. I do not always review this file, though I want to make you aware of it as it can be very useful. When we open this file, we will get a big long list of every single amplicon in our panel. The coverage column, indicated here, lists the number of reads that align to each target amplicon. If you ever see a 0 in the coverage column, that indicates an amplicon dropout, though in this example, we do not have any dropouts. Now we can dig down even further and review the variants found in the sample. There are multiple files that give information on variants, and I've indicated them all here. The VCF files, or variant call files, list the variants within the panel target regions. The GVCFs, or genome VCFs, list all of the possible variant positions and the bases called in those positions, even if they do not differ from the reference, so even if they're wild type. For us to take a look at the variants in our sample, I will show the variant table.txt file, indicated in the lighter blue. I like the way this file is set up, though you can for sure use the VCF or GVCF files to review the variants, whichever you prefer. Opening up the variant table.txt, we see a list of all of our variants. So I'll review the columns here. So first, starting at the left, we see the chromosome and base position, followed by the reference base and the variant, so the alternate or difference from the reference. We have a column that tells us the variant type, so if it's an SMB, insertion, or deletion. Then we have additional columns that give us further information about the variant location and clinical consequences for each variant. We see the gene that holds the variants and additional metrics regarding the variant, including variant coverage or depth. We've just reviewed the analysis results for one AmpliSeq for Illumina DNA myeloid library. Each Amplicon panel will have different expected metrics depending on the panel design and how the libraries are sequenced. Illumina provides expectations for pre-designed panels, assuming an Illumina analysis method is used. For custom panels, the expected metrics will need to be established within your lab. This applies both to evaluating metrics as well as the upstream sequencing optimization, so deciding on the number of samples per run, desired reads per sample, etc. Control samples for which we know the expected variants are very helpful during validation to ensure that we are able to detect everything that we want to detect. You can always reach out to tech support if you would like to review analysis metrics for custom or otherwise Illumina sequencing panels. I've included links to the guides for the software options we have discussed here today, which give additional information regarding the analysis algorithms, the output files, and data interpretation. Now let's do a quick summary of our chat today, and I will include some additional resources. Today, we have reviewed a general overview of Amplicon sequencing. We discussed library preparation methods, sequencing, and data analysis. We discussed 3 key areas for assessing the success of our Amplicon sequencing: evaluating the sequencing run to ensure it meets or exceeds specifications, reviewing alignment metrics for all libraries analyzed together, and we took a deeper dive into the sample-specific metrics to better understand the sample performance and assess the variants called. I referenced this before, though if you would like more information regarding the library prep and sequencing of Amplicon libraries, I highly recommend reviewing the recorded webinar, Amplicon Sequencing Introduction. The Gene Panel Finder and Disease to Gene Finder are helpful tools to identify the most appropriate panel for your upcoming project based on the genes you would like to sequence. And Design Studio is our tool for creating custom sequencing panels. Additional tech support webinars can be found at the first link. We have upcoming webinars, and you can register for these here. We also have recorded webinars on multiple topics, from library preparation to data analysis, which can be viewed at any time. We have helpful support bulletins which include information on best practices for library preparation, sequencing, and data analysis, along with many other topics. We have publication reviews, which feature applications of Illumina Next Generation Sequencing technology, and you can always reach out to tech support via techsupport@illumina.com or by calling us directly. Thank you so much for taking the time to join me today. I hope that this session has been helpful for you.